Again, it is my extreme privilege to turn the call over to your host and moderator today, Diana O'Rourke. Diana, the call is yours.
Thanks, Jeff. Hi, everyone, and thank you again for attending today's online event. As Jeff mentioned, this event will be recorded, and I wanted to let you know that a link to a replay will be sent to you in the next few days in case you'd like to listen again or forward to a friend. Now I'd like to introduce our speaker, Dr. Fabian Burst. Fabian studied technical biology with a focus on bioprocess engineering, technical biochemistry, and bioinformatics at the University of Stuttgart. After graduating in 2003, he joined the bioinformatics group of Professor Jürgen Pleiss at the Institute of Technical Biochemistry in Stuttgart as a PhD student. Fabian's work concentrated primarily on computational studies of antibiotic-resistant proteins and the influence of bound water molecules to protein structure and dynamics. Additionally, Fabian actively worked in collaboration with the High Performance Computing Center and the Visualization and Interactive Systems Group to develop workflows for molecular dynamics simulations as well as novel analysis methods for protein-solvent interactions. After obtaining his doctorate degree in 2007, Fabian continued work at the Institute of Technical Biochemistry on an integrated protein database to analyze sequence structure relationships in beta-lactamases. Fabian joined Certara in 2008 and currently holds the position of Senior Application Scientist, where he helps Certara's clients with informatics solutions to provide improved scientific data access, analysis, and collaboration. Fabian, welcome. I'll now pass the presentation over to you.
Thank you, Diana. Okay. So, talking to researchers across the industry, we often heard that there is a delay to access delay in access to data, and therefore also a delayed analysis of that data. And one reason why this is happening is that people often have to wait, let's say, for an IT specialist to create or update a query that returns the data to the scientists. And one aspect of data access versus data analysis is certainly that it's preferred to spend more time on the side of data analysis, as expensive data is being generated here, so decisions have to be made as quickly as possible. Access to preclinical data in particular, for example, by toxicologists or pathologists, is sometimes hampered by several factors. The data is often unavailable or available only in reports or in other hard-to-analyze formats that could be CRO text files or CSV formatted files. There is a mixture of data that comes from in-house or external sources, often stored in different databases. And the applications that are used to capture and store the data are sometimes limited in the ability how data can be correlated across studies, and have typically a limited query mechanism that doesn't support an ad hoc analysis of the data contained within. Preclinical scientists are forced to review individual study reports to glean learnings from them, so they often spend a large amount of time that, as we have heard, sometimes could be days or even weeks, in order to answer the questions that have been posed to them by their team members or project group or by regulatory bodies. So they have to manually sometimes correlate the data across the studies. And these limitations prevent scientists from obtaining the required data in a timely manner, and therefore cause a delay in answering important questions and prevent making informed decisions. In this webinar today, we're going to present two scenarios from the area of preclinical data access. The first scenario deals with the task of monitoring ongoing preclinical studies, so in particular making CRO data readily queryable and accessible. The second scenario is about enabling cross-study analysis. So, for example, how scientists could respond to questions from his project team or regulatory body questions and we will have one example how a question could be asked to the data to learn something about a structure and outcome relationship. The tool that is being used in those two scenarios is Certara's D360, and I only want to quickly mention that D360 has high-level capabilities of data access, data analysis, and collaboration and sharing. It's applied across the industry in early drug discovery and in the preclinical area, and right now it's used by more than thousands of users across the industry. And if there is one sentence that should describe what D360 is all about, it's that D360 provides scientists the self-service data access, and they're able to formulate their questions largely without the help of any IT resources. Now let's look how D360 applies to the first scenario of making CRO data readily available. So the background of that first scenario is that it's quite common that safety tox studies are being outsourced to CROs nowadays, and those CROs deliver a report throughout the study, either on a weekly basis or more infrequently, and finally there will be a report delivered at the end of a study. The data contained within those files, and there might be just 10 files within one report. The difficulty to analyze the content of the files is that different CROs use different kind of formats, whereas we see that the SEND format starts to be used more commonly, but there are also subtle differences between each CRO. And the intermediate files, in the case you get a periodically update, need to be combined in order to analyze, for example, the trend of a body weight measurement over the course of a study. With regards of how that CRO safety data is finally stored, there might be a structured repository where those files are being uploaded to, but we often came across the situation where these files are just stored in as a set of text files on the local hard disk of a scientist. So from this kind of file access problem, there are some issues that arise, and mainly it's the time that it takes the scientists to examine the progress of a study from those intermediate data files. So imagine that there is an 8-week monkey study that is being conducted at a CRO, and at the end of the first week, a file is being sent to the scientists with the animal data and body weight measurements. And each week, a new report of file is being received that has to be loaded into a spreadsheet application where the data has to be merged with the previous data files. The graphs need to be updated to be able to see trends in observations or any kind of measurements of the animal. And because this is a very tedious data mangling transformation process that takes some time, researchers, as we have heard, only look at a few observations because it's so time-consuming to actually prepare and process these files. Another issue that arises is that different CROs use different terminology or nomenclature, so that makes interpreting or combining data even more difficult. So the key outcome is that there is a delay in the time it takes to make important decisions. So whether you want to monitor whether the CRO is following the protocols correctly, or if you want to understand or see if there are any trend in the data, for example, between a group that get a high dose of the test compound versus a low dose, then you finally might want to adjust or cancel an expensive study based on that available information. And a delay in those critical decisions poses a high risk if it takes too long to answer these questions. D360 has been applied in the framework of this first scenario, basically to remove those technical kind of roadblocks. So it means that D360 is being used for the data delivery and analysis. So the installation of a D360 contains a standard configuration of a safety study data network, and we will see an example of that network later in the WebEx. And users are able to build standard queries. Together with D360 as the point where the user interacts with its data, we have created a scent-based safety data store, which is called the Preclinical Safety store, and that captures the CRO-based data. The administrator can upload the CRO data in scent format or in the CRO custom format, and it allows simple data management. We're now jumping directly into D360 and to see how we actually can query the data contained within this preclinical safety store and do a quick analysis of an ongoing study. We're now looking at the desktop of the dashboard of D360, and with that dashboard here, I have the main part. I'll have my quick access tools, so all those small boxes here are basically predefined queries which I can use or reuse that allow me quick access to a predefined question. On the left-hand side, we'll see options where we actually can then create our own query, and we will see this in the second example. Now, please focus here on this body weight trend widget. This is, let's say, a mask which I created to analyze the body weight progress over the time of a study, and I exposed the ability to actually type in the study number. So I'm interested in actually monitoring, let's say, the body weight of a study that is called S2009-042B. That's the study name or the study number, and we'll retrieve the data. So what's happening right now is that D360 looks into the database, pulls back the data that I've requested in this in that query, automatically formats the data, which we see here in the right in the spreadsheet, and also applies, for example, coloring rules or graphs, which I have defined beforehand. So what actually did we get back by executing this quick search widget? So we have a spreadsheet in which each row actually represents a particular animal. A study group, sorry. Each row represents a study group. The study number here is all the same. There are different study groups divided by male and females. And in those columns with the green headers, we actually have the measurements. These are the body weights of the animals within the group. And we'll also see here that they are ordered by the phase, baseline, dosing, recovery, as well as we see the day of phase. In the remaining two columns, we have the information on the test compound dosage. So obviously here we have a control group, while we have here groups which got a increasing higher dose. The information of the body weight is being graphed in this line chart on the left. So the body weight here is plotted versus the phase and day of phase. So we see the increasing body weight over the course of the study. We see that this is a baseline, then we get the dosing days, and finally the recovery. We see that the female rats usually are much heavier than the male ones. And we can also see that here is a particular group of female rats which show a decrease in body weight towards the end of the dosing phase. And if we click on that line and we have the interactive linkage to the spreadsheet, we see that that's actually corresponding to the study group which got the highest dose of our test compound. So that might be something worth investigating closer. Now, this was an example of how you basically, with one click or entering a study number, can get to a data set with a corresponding graph to monitor an ongoing study or look at trends. Now I want to switch back and show you how the Preclinical Safety Store actually looks like. The Preclinical Safety Store application allows the administrator to upload the file that he receives from the CRO. So in this example, there was a zip file received of the CRO. That's the study we just have seen in the previous example. And want to point out that all examples that you see during this demonstration here are basically made-up examples from our demonstration system and have no relationship to real data. The file format here is in the scent send file format, and after uploading this file, the content of that zip file being extracted and being parsed. And that's an example here that we have our study. The study is now being actively exposed, so the user using D360 can query for that study. We'll see that this has been initially created by a colleague of mine and later on updated. And this is basically the interface that you can delete those files, or if you receive a new iteration after another week of the ongoing study, you upload that set of files and it's being automatically been passed and added to the Preclinical Safety store. Now, coming back and briefly doing a recap of what we've just seen. So in scenario 1, there were 2 parts, and one part was that the preclinical safety data from the CRO has been uploaded into this safety database. But the main part here is that D360 is being used actually to query that database and provide scientists with a quick way to analyze the data. And so this basically leads to that the value of the CRO data that was inside these zip files or CSV files has been unlocked. So what did it provide, or where did D360 actually provide a benefit in that first scenario? So the end user workflow has got much more efficient. So I talked to scientists where they previously told me how tedious it is to copy and paste, first actually import those files into a spreadsheet application and then merge the file around and do the data transformation. So there's faster decision, so time is being saved, and the time from receipt of the data files until the actual analysis and the final decision making is greatly shortened. The Preclinical Safety store also allows to handle different terminologies, and being able to have that simple access to the data allows a much better trend analysis. You can now easily replace the body weight by any other measurement or observation, and instead of having to regraph that in a spreadsheet application, you can now basically look very quickly at all the, 35 observations that are being delivered by the CRO. Important, I want to point out that in this case where we combined D360 with that Preclinical Safety store, D360 is flexible in terms of its data sources. So if the CRO-based data is right now stored in another, let's say, a structured storage space, then D360 is finally happy in being connected to that existing database and query the data from there. So it's not tied to the Preclinical Safety store. Moving from the ongoing study monitoring or the basic graphing of CRO data now to a little bit more kind of scientific data analysis and ad hoc workflow scenario. So the background here is that this is modeled after a real case study or real application case. We work with a customer. Safety studies are being conducted on worldwide basically and are also outsourced to CROs. All the safety data is already captured in a commercial system. So that's used by the toxicologists and pathologists to do study monitoring. It's a validated system which is used for reporting, and to make the data a little bit more queryable, a data warehouse had already been constructed from the safety data. So the pathologists and toxicologists are responsible for either diagnosing adverse events in safety studies, but they're also responsible for answering the questions that come up from a project team or regulatory bodies. That all sounds good so far, but there wasn't a big issue with examining data across different studies. It was not possible in the current setup. And if we look at the pathologist's workflow, so that person receives a microscope slide with some kind of biological sample. The person is actually analyzing that sample, storing its finding in the commercial system. That's finally a database. And he repeats that for a lot of those slides, for a lot of different kind of organs and observations. But the final outcome of all that, of this procedure, is that a study report is being generated that then can be handed over to the FDA, for example. Now in the case that the project team has a question, or the FDA comes back with a question because they read the report very carefully, usually the analysis of or the task you have to do to respond to those questions involves looking at the baseline data for control groups. And the creation of such a data set usually means that you have to export all the relevant studies, for example, all the doc studies that you have done in the past, and that means you get a lot of individual doc study reports. Then you extract the control group data from those reports and put them in a spreadsheet application where you have to merge them all together to finally derive or distill out an answer that you can send back. And this is a very, very time-consuming process that, as we have heard, can take really days or weeks. So I mentioned the data network that is being used in D360. The data network means that we analyze the data that is in the safety database and basically cast a data network with the entities that are of interest in this disciplines and here in the preclinical area. So that means that we'll create a data network where we have terms like a study, which consists of multiple study groups. Each study group consists of multiple animals, and for each animal we have a lot of study observations and measurements. But each study group and each animal might have gotten a treatment, which might be a certain compound or vehicle, and scientists are with D360 now able to basically look at this data network and ask questions from all sides of their network. So show me a certain observation for all animals that got a certain treatment, or just give me a measurement for all the control groups, and so on. So this supports a wide variety of user workflows, for example, the aforementioned cross-study analysis, but also many more, like designing study from historical data or the logistical study monitoring. But in fact, instead of pulling out all the individual reports, extracting all the historical control group data and mangling around with it, a cross-study analysis can now be performed basically in minutes rather than in weeks. We'll come back to this data network later in the presentation and see how this, for example, can be extended. I would now like to come to the next demonstration. We have two examples in the cross-study in this second scenario. And the first one, I call it the renal finding in dogs. So what is the situation here? So after reviewing our trial data, the FDA is concerned that there is a 4% of our dogs display a phenomenon of renal basophilia compared to 0% in the control group. And now the FDA wants to ask or wants to know from us whether this is a significant finding or not. So the question that the pathologists or toxicologists in the scientist group now has to answer is, show me all the control group animals that are dogs and their respective renal findings. And now I need to determine what percentage of those dogs show renal basophilia, and probably if we can break down this by study and by gender of the dogs. So I will now switch again to D360, and instead of running, closing this data set here, instead of running one of those predefined quick access widgets, we are now actually going to build a query in D360 ad hoc to answer this question. Now, we choose to build a new query, and this is going to be an animal level query. Remember the data network where we could basically ask questions from various kind of levels? We now ask a question on the animal level. So every row in our result represents an animal of the study. Okay, we are now in the query building interface. On the left-hand side, we see what we call the data catalog. This shows all the information that has been configured to be accessible by D360. Now we're interested, of course, in animals, so we would like to get the animal number in our result. We, of course, want to know the animal species, and as we have to defend our dog study, we are interested in only receiving results where the animal was a dog. And we would like to see the gender of the animal. You see that the species name here has highlighted in a bluish color. That means this kind of query band is being constrained. So right now we would return all animals that are dogs. Of course, we want to focus here on control groups. So we'll add a few more things from the study groups folder. For example, the control group item here, and say, yes, the control group, that should be a control group. Now we have two constraints in our query, the species name dog and the control group equals yes. And we want to see the control group name. Okay, so now we get all control group dogs together with their name, gender, and so on. We probably also want to know in which study that dog has been used, so we go into the study level, study folder, and just include the study number into our query. Now we're finally there. We probably need the most important thing, and that is the renal basophilia finding. So now where can we get this? So we have an in vivo results folder here, where basically broken down all the results that are stored in this database, and I could assume that the kidney finding is somewhere in the microscopic observation section. So, but I'm not really sure. So why don't I just use the search functionality, typing in kidney here, and I'll see that there's a kidney finding in the microscopic observation folder, and I'm just using that finding here. And we do not constrain this, so the idea is now to run this query, and the query should return all dogs which are part of control group and their respective kidney finding. So now let's run that query. Okay, already done. We see that we have returned 247 animals. We see the study animal number, species, the gender, control group, yes, so we don't need this. We know that we asked for control group, so we get rid of that column. We get the study group name and the study number. And we get the kidney finding, and we see that there were animals without any kidney finding. We see an animal here with a basophilia finding. So now let's see how many animals within those 247 animals actually had a kidney finding of basophilia. So we're using here the select record tool, and we'll just we'll add this, and we'll say the kidney finding should contain the fragment baso for basophilia, and we'll just select this. Okay, so this now has selected all the rows where the kidney finding contained basophilia, and we see that we have around 14 out of the 247 dogs present with basophilia. And if we just pick out the calculator and do the math, we'll see that this equals to a little bit more than 5% of the control group dogs showing a basophilia finding. So let's return to our presentation. So the result of our quick ad hoc analysis, which I hope didn't take any longer than a couple of minutes, is that we have a little bit more than 5% of all our historical control group dogs demonstrating renal basophilia. So the 4% level that we've seen in the non-control population probably is not significant. And if we would have done a more in-depth analysis by adding, for example, a few histograms, we could have seen that it's more likely in female dogs versus male dogs. And the screenshot I'm showing here is actually the same data set in a different kind of format with a few more viewers added, and it would be a couple of minutes to add those views and the coloring. So this really shows that we're able to get insight into this example here in this use case very quickly. So just to mention that previously this would have been involved extracting all the control group animals from all studies, dog studies, and merging this in a spreadsheet to be able to do this analysis. So this enables scientists to provide a fast response, and a fast response is important because any delay has a potential impact on the approval process if the answers from the FDA are not, or the questions of the FDA are not answered on time. Moving to the second example in this scenario, this is more a question that a project representative might have been tasked to look at. So it's more looking at the bigger picture on the structure-outcome relationship. So imagine the question might have come up, I need to know how frequently we see nerve damage in any kind of animals, and could this potentially correlate to the compound or the test compound that we administered to those animals? So the question that scientists would need to ask to its data sources, show me all animals that have displayed any nerve damage finding, and we want to break this down by study, test compound, species, and, for example, test compound structure. Now again, switching back to D360, we are using, again, one of those quick search widgets on the dashboard. This widget here is querying animals for microscopic observations, and we are looking for microscopic observations in the tissue type nerve. We're going to execute that widget, and this widget is set up in a general way so that, for example, the tissue type could be changed. So this is a widget that can be shared amongst colleagues and can be easily changed also to answer different kind of questions. Now, we see a little bit kind of a different result here. We have the spreadsheet, but we also have like 3 histograms which have been added. So as well here, after retrieving the data, automatic kind of macro steps are being applied. We get the study number back. We get the duration of the study, where it has been conducted. So there's studies in Asia, a study in the US. When they have been conducted, we have a mixture of rat and dog studies. We also see this in the histogram, which we done on the species column. So around 50 rats and a little bit more over 5 dogs displayed obviously a nerve damage. And we see what kind of nerve damage observation has been detected. So there might be a degeneration or infiltration or a mixture of both. We also see the test compound here, so D360, as I told before, is also used in the early discovery area, so it has a chemically aware spreadsheet. We see the structure that has been administered to those animals and together with the dosage of that test compound. Now, the analysis that we could do from this spreadsheet here is we can see which compound here shows the most nerve damage findings. So there's obviously one compound here that has been quite often has shown a nerve finding, so over 10 times. We also see that the breakdown by study number, so we see that there's only one study which obviously used that test compound which had a high level of nerve findings, and we see the breakdown by species. Now, with the chemical structures being added, we can use and leverage some of D360's chemical analysis capabilities, and we can do a clustering of those structures. So we want to see how similar are those structures. So what I'm going to do now is I'm adding another viewer, which is basically a clustering algorithm that places the chemical structures onto this plane here based on their similarity to each other. So dots, which are close together, represent similar chemical compounds. And we see that we are talking about 4 kind of clusters here. So there seem to be 4 clusters of chemical compounds associated with nerve damage in all our studies. And if we hover over the mouse over those points, we'll see that those are indeed very similar looking compounds. Now, if I'll focus on those 5 compounds here, we actually see that these compounds, which all look very similar, are actually responsible for over 60% of the nerve damage finding. Can compare this with the number of selected records here. So one of our immediately outcomes would be that there seems to be a set of related chemical structures which have a high potential for nerve damage. By looking at the others, we actually can see that there is a set of compounds that has nerve damage in both rats and dogs, so that shows nerve damage in the 2 different species. So with that, I want to jump back into my presentation. So the result here is that nerve damage is, and compared to the amount of dog studies that we have done, we didn't bring in let's say, the animals that did not have a nerve finding, but we could have easily done that. So nerve damage is not very prevalent in non-control groups. However, 60% of the nerve damage finding could be traced down to a group of similar compounds. So as a user, you really have control of what kind of questions you ask to your data. You'll be able to access all your data, and you're not tied to a kind of black box of a predefined question. This example here incorporated structural chemical data, and therefore also demonstrated the ability of D360 to access data across different disciplines and to tie together preclinical safety data with discovery data. So looking back at those 2 scenarios which I did present, what were we able to achieve with D360? So first, scientists were able to answer questions faster, usually without a disruption of their ongoing work, and the burden of tedious manual data manipulation and copy and pasting, which is a great source of errors, was taken away from them, so therefore improving or leading to improved decisions. So secondly, the agile data access and the analysis capabilities in D360 can encourage scientists to explore their data a little bit more and lead to an improved data interpretation and in study design. So users can, for example, investigate new scientific concepts like look for vehicle toxicity. Very important, the D360 offers a self-service kind of data access. So all these kind of questions can be answered by the users and do not require the involvement of any IT support. So that's also important because rarely the same question comes up twice, so IT constantly would have to adapt to new questions. And finally, getting to the data in a more streamlined fashion without the risk of introducing those errors with the manual data curation. The users can spend more time on the data analysis side compared to data retrieval. And this, together with the possibility to ask these ad hoc questions from all ends of the data network, enables to do and answer exciting questions which were not possible before, and that basically leads to a much better scientific outcome from the data. I promised to come back to the data network towards the end of my presentation. So as an outlook, how could the data network that we used in the preclinical space could be expanded if we would have access to all the data which is available in our organization? So what kind of questions could we address? So if we recall that data network, which we cast in the preclinical area, we see that it is expanded here by sections like compounds or in vivo data or TK parameters. So as we've seen in the second scenario, adding chemical structures from a discovery database would allow to ask interesting questions like structure-outcome relationship. TK parameters offer the possibility to investigate exposure-activity relationships, for example, and having access to assay data from the discovery area would allow to ask questions like, are there any biochemical assays or enzyme assays which could be used for predicting any certain kind of outcome or observation. So with an expanded network like this, that would really mean that there is that D360 can span a bridge between the silos of discovery and preclinical data. So to summarize, D360 is in use in the discovery area, and if you're interested in actually looking at D360 in action in the discovery area, I would like to point out that there's a recording of the previous webinar available on our homepage. In this webinar, we've shown that D360, how D360 is able to unlock the value that is in the preclinical data with two scenarios, with the first accessing and analyzing study data from CRO studies, and the second scenario was a more kind of scientific workflow, ad hoc questioning, cross-study analysis. All these questions were supported by the same data network that has been used in all these scenarios. And finally, again, D360 allows the scientists to ask any kind of questions to their data without the constant help of IT support. And with that, I conclude my presentation and I hand it over to Diana for Q&A. Thank you.
Thanks, Fabian. We do have several questions that have come in, and so we'll jump right into the Q&A then. The first question is, can you integrate with other statistical tools like JMP?
Yes. So, for example, especially if you mention GMP, there is a built-in integration into D360 as well as for other analysis tools. And if it's not, then they can be added based on request.
Okay. Next question, is D360 customizable to other data sets such as in vitro, etc.?
Yes, so basically the D360 can be configured to access all kind of data sources, and it's about building the data network that we've seen here in the preclinical area. But yes, so it's not tied to the preclinical area as it's been used, for example, in discovery as well. So it's a matter of configuration. Yeah.
Okay, great. You mentioned a database for CRO preclinical results. What formats of data can be added to that system?
So that preclinical safety store that was established here can load scent data and also supports some custom formats provided by the CRO. And so we add support for new formats as it's needed for our customers.
Okay, great. How do you deal with terminology differences when data comes from different CROs?
Yeah, so especially in the with this preclinical safety store, there is a terminology module that allows to alias or allows aliases to be associated with preferred terms, and also scent control terminology. It could be loaded into this system.
Okay, just want to remind everyone, we do have a few more questions in the queue for the Q&A. There is still time to submit your questions, so just use the Q&A panel for that, and remember to submit to all panelists for us. So there is still time to submit questions if you have one that you'd like to ensure gets answered. So next question, Fabian, does it matter what kind of databases our preclinical data is in?
No. So, so as I mentioned, D360 can connect to a variety of data sources and or databases and also web services. So, but it's important that the data is in a performant data source or in some kind of structured, stored way. So Excel sheets or text files do not work very well. So this is exactly why we have developed the Preclinical Safety Store, to get the formatted text into a more searchable and queryable format.
Okay.
So, yeah.
I'm sorry, did I stop you before you were finished?
No.
Okay. Okay, great. How does the application get connected to the databases?
So that is basically a standard configuration task that is done during the installation or deployment of D360. So there's a configuration tool to do that.
Yeah. Okay. You've described the potential of a company to reply for a question from a regulatory authority, but I'd like to know, how does it work in the other way around? Since we do receive dossier, it's on a PDF format, what is the issue for us?
I'm not sure whether I understand that question correctly, but so if a PDF is received as a report, basically the information from that PDF would have need to be extracted and made searchable. So usually what we've seen is that customers that until now receive data in PDF report formats are now requesting from their CROs to get the source files with the individual data so they can make them searchable through D360.
Okay. Have you linked the preclinical data with earlier phase biological data at this point?
Yes. So what we've seen in the demonstration system here, of course, was built for demonstration purposes, but we actually, in fact, have a couple of customers who are starting to do this, and we develop the workflows with them, exactly the workflows they are interested in to run in D360. So yes, that's actually something that exists in reality.
Okay, great. Well, Fabian, thank you. You've done a great job of answering the questions that have been submitted by our audience. This does conclude the questions that we've had submitted through Q&A. So this does conclude the question and answer session. I want to remind you that a link to a replay of this webinar will be made available on the Certara website within the next few days. And we will be sending an email to you with a link as well, so you can replay or forward to a friend. The current slide provides information on how to contact us with questions about today's presentation or to get in touch with our sales team about any of Certara's products and services. On behalf of Certara, I want to thank you for attending today's presentation. This concludes today's webinar, and you may now disconnect.